So far we have seen the influence of substrate concentration, inhibitor concentration or activator concentration on the rate of enzyme catalyzed reaction, and they very strongly influence the catalytic behavior. Now, besides the substrate inhibitors or activators, two major environmental parameters which form or which constitute a major environment, bulk environment for the enzyme catalyzed reactions are pH and temperature. And today we will be looking at these environmental the parameters and how do they influence the whole enzyme catalyzed reactions. Now, as you know that enzymes are primarily very complex molecules, and we had also looked into the complexity of the situation as regards to their catalysis, as regards to their specificity, and the whole effect of pH and temperature therefore is not based on a single interaction, like in the case of substrate or an inhibitor, which is usually the effect on the reaction rate is usually based on dynamic enzyme, their formation of dynamic complexes with the enzyme. In the case of pH and temperature, the effects are multidimensional, and if you look at precisely, just to come first of all at the pH effects, you will notice that the pH influences enzyme reaction rates or enzyme reactions in general in a number of ways. The first and probably the most common feature could be the shift in equilibrium. A number of enzyme catalyzed reactions that are reversible in nature, primarily they can be affected strongly by the pH, and of course in those cases, particularly the reaction like oxidoreductases. If you take for example ethanol oxidation by alcohol dehydrogenase forming acetaldehyde plus NADH plus H. If you consider such a reaction, you will notice that the concentration of the hydrogen ions at any given pH can be regulated by the pH of the environment, and so by taking a buffered reaction mixture at a higher pH, the concentration of the hydrogen ions in the reaction mixture can be maintained at a very low level, and the reaction can be driven in the forward direction. And similarly, if you reduce the the concentration of the or the pH of the buffer in which the reaction takes place, you can also bring down the equilibrium constant of the reaction significantly. Now, this shift of the equilibrium is again, although not directly related in any way, but is influenced or must be considered in conjunction with the rate of the reaction. Now, for example, if you consider this reaction, although this reaction may have an optimum pH of say for example 7, that means the rate of reaction is maximum at pH 7, but at pH 7 the reaction may not be driven very largely to the forward direction. The reaction may stop as soon as some concentration of the product and hydrogen ions are built up. Now, to be able to drive the reaction in the forward direction, you need to adjust the pH so that the concentration of hydrogen ions can be reduced, and therefore the reaction can be driven, but the reaction might proceed at a very slow pace. For example, if you consider let us say pH 9 just as an arbitrary pH value. Now, at the pH 9 the concentration of hydrogen ions will be 100-fold less, 1/100th of at pH 7, and therefore you can drive the reaction as far as the equilibrium constant is concerned, but at a very slow rate, and obviously the shift in the equilibrium must be considered in terms of the rate. So, and so pH optimum or the equilibrium constant, they are two issues which are to be considered independently. And now the second issue which is important in the case of pH effects is the stability of the protein conformation. As we all know that proteins retain their native conformation within a limited pH range, because electrostatic interactions which are a major force, non-covalent force involved in the conformation of the enzyme molecule. So, within a limited pH range at which the right kind of amino acid residues are ionized, the native conformation of the protein is maintained, and if you shift that pH, very often the gross alterations occur, and what we often call that a denaturation has occurred. That means, if there is a irreversible change in the conformation, the enzyme is considered to have been deactivated. So, basically we have seen the stability behavior of enzymes even in our earlier stabilization studies, where we saw that a two stage model is the most accepted model for enzyme, which consists of two steps: native protein going into a unfolded form and then irreversible inactivation to an active enzyme. Basically, while the first step is reversible, the second step is irreversible, and always the approach to stabilize, and now similar effect is noticed even for the pH. Initially, if the pH alteration is mild, if you go beyond a certain level of pH change, you might observe only reversible change, and therefore the enzyme can be brought back to the native conformation. But if you further change the pH, it might undergo to a irreversible inactivation, and which may not be possible to bring back. The third role is in which the hydrogen ions play a role for either inhibitor or an activator, and obviously wherever we are considering the involvement of ionizable substrates or the products that are formed which are ionizable, the hydrogen ions will play as a role of a inhibitor or an activator. And of course, the fourth role of pH, and probably one of the most important role which pH plays and which directly influences the rate of reaction, is the ionization of amino acid residues on enzyme active sites. We know that on the enzyme active sites a number of amino acid residues are present which are charged, and these charged residues will ionize as a function of pH of the bulk environment, and therefore it is required that they are at a particular ionization state in which the enzyme offers the maximum reaction velocity, and essentially the effect of pH is essentially linked to the state of the ionization state of the enzyme active site. That means, if there is a particular state of ionization of the active site or the amino acid involved in that, whatever fraction of the enzyme is present in that form, that fraction will constitute the catalytically functional enzyme. Rest of the enzyme concentration will not be useful, and therefore this ionization state will essentially affect the maximum reaction velocity of the enzyme, because ultimately you are affecting the total enzyme concentration, effective enzyme concentration, which is in the true ionizable state of the system. The last effect is due to ionization of substrate. Although many of the substrates which are used in enzyme reactions may be neutral, very often like ethanol or any other non-ionizable substrate, but many of them also contain carboxylic group or hydroxyl group or amino group, and they will all ionize also, substrate also will ionize, and depending on the true state, ionization state of the substrate which is accepted by the enzyme at the binding site, the fraction of the substrate which will get bound will determine the pH effects, and essentially the ionization of substrate essentially will influence the Km value of the enzyme. The Km value will be altered because the apparent concentration of substrate which we are providing is not the true substrate concentration which is interacting with the enzyme, and it may be lesser than that because the ionized state which is catalytically acceptable to enzyme is less than that, and therefore the pH effects must be looked into all these parameters or all these effect in a comprehensive manner. And of course, practically it may be difficult to consider all the effects in one go, because sometimes they may be counteracting, and so the net effect what you notice is a very complex phenomena. However, to understand the behavior and to derive certain meaningful conclusions, one may create or design experiments in which only one of the pH effect is considered prominent, and in fact the pH effect studies have contributed a lot to understand the identification of the amino acid residues on the active site, in conjunction with, again not alone, in conjunction with the chemical modification studies. Very often a common technique to study the amino acid residues or identify the amino acid residues at the enzyme active site is by modification, chemical modification of the different functional groups that are present. In addition to that, their effect on the ionization behavior and the ultimately the rate effect, and putting both the things together can give you a confirmatory result or conclusion on the identification of the amino acid residues on the reaction rate. Now, essentially you may notice that we will essentially consider today mainly the ionization of amino acid residues on enzyme active site, which is primarily a major factor which influences the typical reaction profile that we are familiar with of the effect of pH on the enzyme. Now, you often end up getting a variety of profiles when we study, let us say pH, when we study the enzyme reaction rate as a function of pH, we get a variety of profiles. Now, for example, in the first case in the diagram in the part A, you notice that the effect of pH is shown against the percentage rate of the maximum value, that is maximum value means the optimum pH and the optimum or the maximum at which it shows, and with a single ionizable residue on the active site. When there is only one ionizable residue with a pK value of 9, you get initially at a lower pH value the enzyme activity remains constant till when you go to higher pH value the rate falls, and subsequently at an extreme value it comes down to 0, and we will take the determination of pK values and their role. Now, in the second case you notice that it may be a much broad based pH profile, very typically a profile like this, which essentially shows maximum value over a pH range. Of course, not a very broad range, but at least in 1 or 2 units it exhibits. Now, this reflects probably the presence of 2 amino acid residues, ionizable amino acid residues, one of course acidic, one basic on the enzyme active site, and the profile is like this, and with the 2 amino acid residues, one of pK 3 and one of pK 10, just typical profile which you get. Most commonly you may end up getting a profile of a very sharp maxima, and we are very often familiar with a typical terminology called bell shaped curve, and you have a very sharp pH optima which is considered at the peak of this. Now, essentially such a situation is a special case of this broad profile, in the sense that here the pK values of the 2 ionizable groups are distinct apart, one at 3 and other at 10. So, their ionization does not really influence each other so much, and when one of the amino acid is ionizing, the other is not in a very highly ionized state, and therefore you get a broad pH and there is no problem. But in case the pK values of the 2 ionizable groups are close enough, that means that the ionization of at a particular pH, the ionization of one amino acid residue does also ionize to a of course different extent the other amino acid residue. In that case they will tend to look at it if both the results are merged, both the ionization results are merged, and you get a sharp profile, and because here the pK values of the 2 amino acid residues are 7 and 8, which are pretty close with a difference of. So, that is the kind of scenario you get, and you can really get, in fact, as a matter of fact, these are again I would say comparatively simplistic situations. As a matter of practice, in many of the enzymes for practical applications, you might end up almost like a profile something like this, which is consisting of not 1 or 2 amino acid residues, ionization of 1 or 2 amino acid residues, but more than 2, maybe 5, maybe 7, maybe 6, and essentially the net pH profile, pH and percentage maximum reaction velocity may be a sum total of all the effects, and they can be broken down into segments, linear segments, which will each linear segment will represent the ionization of one of the amino acid residues. So, basically I think the effect of pH on the reaction velocity must be considered as a parameter on the rate of reaction. Now, let us take the most simple case, the simplest case, of course, the effect of pH, and that the simplest case, as you will agree, will constitute that if we assume, like in the case of the first case, that the enzyme possesses only one ionizable amino acid residues, and it can be considered, the ionization of the residue can be considered as EH going to E minus plus H plus. That is, the neutral enzyme, which is protonated enzyme, can dissociate into the negatively charged enzyme and the protons, and of course, let us say with the ionization constant of K. If now under these circumstances, the concentration of EH will be E minus into H upon K, and of course, the total enzyme concentration E naught will be your EH plus E minus, and therefore, this will be your EH plus K into EH upon H plus. Again, you can take EH as a common factor and 1 plus K upon H, and of course, therefore, EH concentration will be E naught upon 1 plus K upon H plus, or in other words, you can also simplify E naught into H plus upon H plus K. Now, essentially we assume that EH is the catalytically active form and not the negatively charged enzyme, then the substrate will bind only to EH, and EH plus S will go to EHS and followed by EH plus P. If we assume that EH is the catalytically active form, in that case the VM, the maximum reaction velocity will be that EH will, all the enzyme is present in the form of EH, and will be essentially K into E naught into H plus upon H plus K. Is that right? Now, the BM will be essentially, if all the enzyme is present in the form of EH, so therefore, you multiply K, the rate constant K here, with the concentration of EH. Now, essentially you look at that in this case what has happened, if all the enzyme, the total concentration is E naught, if the total concentration, total enzyme is present in the form of EH, that is your VM true. That is essentially the maximum reaction velocity which one can achieve if at a pH, at some pH, when predominantly all the enzyme E naught is present in the form of EH, that is the concentration of E minus is negligible. That is a true VM actually, and this upon your 1 plus K upon H. Essentially you arrive at such kind of a relation whereby you see the VM is affected by a factor 1 plus K upon H. Of course, in such a case, if you plot the profile of pH, which is negative logarithmic of the hydrogen ion concentration with log of VM, you end up a profile like this. Now, if you can even get from that, if you take logarithmic of both the sides, log Vm is equal to log Vm true minus log of the term in the parenthesis, and if you just consider the extreme conditions, that is when your hydrogen ion concentration is much greater than the ionization constant, and or in other words the pH is smaller than pK, then your log Vm is equal to log Vm true. That means it remains constant at a value of pH which is lesser than pK value of the ionizable residue. The Vm remains constant and at its original value. Only when the hydrogen ion concentration becomes smaller than K or pH becomes greater than pK, the log Vm is equal to log Vm true minus log K plus log H. The hydrogen ion concentration will influence, and essentially the Vm will be decreased, and you get a downward fall. And essentially if you just extrapolate these two lines, this is the pK of the ionizable, and at the point where the pH and pK are identical, you will get a point of intersection at which the two lines will merge. So, therefore, that gives you a, on the other hand, if you consider the other alternative, that as we noted that if instead of EH, if E minus or the negatively charged enzyme is the active form, and that is another option available to us, if the E minus is the active form, not the neutral enzyme, in that case the rate profile, although analysis will be identical, but the rate profile will be different. In the case of E minus is the active form, the rate profile will be your Vm is equal to K E naught K capital K upon H plus K, and you can again analyze the same in the same way, and the profile obtained will be your reverse of this, and as you noted you can go again, you get pK value. So, the only difference will be the direction of the pH effect will be changed, and obviously the analysis will remain on the identical lines. Now, that is of course a very simplistic situation when we have, and very often you will find rarely an enzyme which really gives you a situation. It is only to appreciate and understand the effect of the ionization of the amino acid residue on the reaction rate. But if you just take a little more realistic case, which again in many cases may be true, that instead of having one amino acid residue, we have two amino acid residues or two ionizable residues, rather not two amino acid residues, but two ionizable residues. There might be more than two other amino acid residues which are involved in the active site, but they may be non-polar, and therefore if they are involved two ionizable residues, let us say one containing a carboxylic group and a amino group, and definitely if you just add acid or base to or change the pH, the ionization state of the two residues will change, and if we assume that both groups being in the ionized form, if it is the active form, then you can see the effect of addition of acid or base in this equilibrium, and if the middle thing is the ionizable form, you can also predict that the left hand side form of the enzyme ES2, which has a more reduced form than the EH, and then EH is more reduced than E, and therefore, and if this is the EH which is the form which is catalytically active, that means which binds to the EHS, which binds to the S substrate and forms the product. Now, to analyze the system, we consider that there are three dissociation constants involved. One is the ionization of the two amino acid residues. If we represent Ka and Kb as the ionization constant of the two residues that are involved, and the third is of course we are taking Km, which is the dissociation constant of the enzyme complex with the substrate. We define it analogous to a dissociation constant as Km. Now, obviously, in such a case the typical profile that will be obtained of pH will be a well shaped curve with a pH optimum at the middle of it, which we consider as the typical profile on BM. Now, if you look at a bit of analysis of such a system, you will notice that you can write the three different equilibrium and get the concentrations of individual enzyme species that are present in the 3 state. Now, let us consider the first one whereby in the case of EHS, that means the interaction of the EH with the substrate, we can write the concentration of EHS will be equal to EH into S upon Km. Now, I am omitting the parenthesis to denote the concentration term. Essentially, these terms represent the concentration terms. Then similarly, if you look at EH2 will be your EH into H plus upon Kb, and E will be your K into EH upon H, just based on the equilibrium dissociation of the 3 species. And of course, our total enzyme concentration E naught will be equal to free enzyme plus EH plus EH2 plus EHS. Basically, the total enzyme is present in 4 different species: the free enzyme, the partially reduced, fully reduced, and the substrate bound form, of course, which is involved, which is binding only with the EH. And therefore, we can just write, substitute the values EH 1 plus Ka upon H plus H upon Kb plus S upon Km. Now, therefore, from this a bit put the expression in the term for EHS. Your EHS will be your E naught into S upon Km whole divided by the whole term in the parenthesis, that is your 1 plus Ka upon H plus H upon Kb plus S upon Km, this whole term, or your reaction velocity will be equal to your K into EHS, that is the concentration of your enzyme substrate complex, which is bound the doubly ionized enzyme complex, that is your K into E naught into S upon Km 1 plus Ka upon H plus H upon Kb plus S. Now, here if you notice as that the whole effect of the change of pH, that is the hydrogen ion concentration, is influenced by this parameter in the parenthesis. The parameter in the parenthesis effect influences or depicts the effect of pH on the reaction velocity. And essentially, if you consider this, now this parameter will pass through a minima as hydrogen concentration or the hydrogen ion concentration is varied, and now you can get the minimum value by simply differentiating it with respect to hydrogen ion concentration. And if you consider this function, if let us say we denote this function as f, your df over dh will be equal to 1 upon Kb minus Ka upon H square. If you equate it to 0, you get the 1 upon Kv is equal to Ka upon H square. So, therefore, your hydrogen ion concentration which gives you this function f to be a minimum. When this function will be minimum, your reaction velocity will be maximum, and that will be given by under root Ka into Kv, and therefore, you get the minimum value of this function. This is not the minimum hydrogen ion concentration, but is the value of the hydrogen ion concentration as which the value of this function f is minimum is given by that, and obviously the rate will pass through a maximum when this function is minimum, and at this concentration of hydrogen ion concentration, the reaction velocity will pass through a maximum. So, this is essentially you can use this expression to denote the effect of K, the effect of ionization constant of two amino acid residues on the reaction velocity of the enzyme catalyzed reaction. Now, I think in terms of if you can increase on the complexity by taking more and more number of ionization. Now, not necessarily that there will be always one negatively charged and one positively charged amino acid residue. There might be more than one of each of the type of. There might be two negatively charged, there might be one positively charged, or there might be two acidic and one basic, or more than that in the case of a particular enzyme. The only difference you will notice in that case, the pH profile, the pH optima profile will become not a very sharp well shape, but can be broken down into different stages or segments, which can each of the segment representing the ionization state of 1. And also it will be important to notice that the sharpness of the profile of the pH versus reaction rate will also depend on the proximity at which the pK value of the different amino acid residues lie. And very often, just by a pH profile, if the number of amino acid residues involved are not very large, one can judge essentially the likely number of amino acid residues involved, and with and knowing the pK value of different amino acids that are known, that are likely to be present, one can make a judgment whether a particular ionizable amino acid residue is present or not. And so, that is what is the effect of pH as far as the ionization of enzyme active sites are involved. The second, the next stage, which is again very important, is to look at the ionization of substrates. Because it is not only important that the enzyme may ionize, it may also be very commonly feasible that when you are handling the charged substrate molecules, and very often even many times the protein itself is a substrate which is a polyelectrolyte, and it will have many functional groups which will be ionizable. In those cases the binding of the substrate to the enzyme will depend upon the ionization state of the substrate. And of course, there might be depending on the number of charged functional groups on the substrate, the situation might be more and more complex. But just to consider two simple model cases, let us consider a substrate which in its unionized form is catalytically active, or not catalytically active, is able to bind to the enzyme for catalytic function, because substrate itself is not catalytically active, it is only enzyme which is a catalytically active component. And if suppose we assume a simplistic case of the neutral substrate going, but the substrate can ionize and give you a protonated form of substrate, for example at the ionization constant of K, assuming only one ionizable. Now, you can arrive at the rate expression for such a system as Vm into S upon Km 1 plus H upon K plus S. Now, you will notice that in such a case the net effect of the ionization of substrate essentially is a increase in the Km value. You have the Km value has modified to the true Km 1 plus H upon K, and the system or the expression is analogous to that of competitive inhibition. That means, in such a case the hydrogen ions act as a competitive inhibitor for the substrate. The neutral substrate, unprotonated substrate is the reactive form, is the form which is acceptable to the enzyme. Whatever substrate, part of the substrate which has protonated, got protonated by binding with the hydrogen ions, becomes a dead end complex, as happens in the case of a competitive inhibitor, and therefore you get a similar profile which gives you a Km value. And you can also analyze now here the Km apparent, if you consider K dash m as a Km apparent, varies with the hydrogen ion concentration. So, the effect of pH on the Km value can be noted to look at the pattern of inhibition as far. In the second case, if we assume that it is the charged form, is the protonated form which is catalytically acceptable to the enzyme function, now in such a case, although the system, the analysis will remain the same, only the form of the reaction velocity will change to, again there will be a difference in the Km value, you will again get an apparent Km, but the magnitude of apparent Km will be slightly different and will be given by Km into 1 plus K upon H plus S. So, essentially you will notice that if you analyze the two apparent Km terms, in this case this one, and in this case Km into 1 plus K by H plus, you will notice that only at extreme values of hydrogen ion concentration there will be difference in the behavior of the enzyme, otherwise they will remain almost a similar kind of a behavior. By increase, and in both the cases you will notice that the ionization of substrate influences the Km value of the enzyme, and that is increased, and this increase is in magnitude dependent upon the fraction of the substrate which is present in the form of the substrate molecule which is acceptable to the enzyme and ionization substrate. And similarly, if there are more than one ionizable ionizable groups in the case of a substrate, a similar analysis can be arrived at to for considering the effect of.
So,
yes.
So the second expression that we have derived, won't S be replaced by S is positive?
Which one? You are saying
V
this
naught
one?
is equal to Vms Km plus 1 upon K plus H negative.
No, essentially in this case, yes, you are right, we should consider SH as the SH positive, because that is the form of substrate concentration which is essentially the total substrate concentration for all practical purposes. You are right. Now, the second part of our considerations today are the pH effects. So, we have seen essentially so far that the pH effects are quite complex, and all the parameters that are influenced by pH cannot be considered in one go. They need to be considered in isolation one by one, and depending upon the information we want to collect. Sometimes the information required is only to understand the optimum pH value of the reaction. So, the life is very simple, that you conduct the enzyme catalyzed reaction in a buffered medium with different pH values and get the reaction rate and use the one which has the maximum value. Sometimes you want to know identify the amino acid residues at the active site, you need to make analysis of the enzyme reaction and the pH enzyme rate profile, detailed analysis to get the pK values of the amino acid residues involved and thereby make deduction about which need to be confirmed again by chemical modification studies, because such analysis can give you an indication, but may not be able to give you confirmed data. The second environmental parameter which is very important is temperature. Now, like pH, temperature is also a multidimensional parameter which influences different aspects of the enzyme catalyzed reaction. Probably the very first step at which the temperature influences is the interaction of enzyme substrate complex, an equilibrium process. We know it is a reversible equilibrium process, and essentially the key parameter which represents the ES interaction in our case, as we have noted, at least for typical Michaelis-Menten kinetics, is the Km, Michaelis-Menten constant. Now, the temperature will influence the effect on Km, and the study of effect of Km, effect of P or temperature on the Km is a very interesting issue, because it gives you a lot of insight into the thermodynamic aspect of the enzyme substrate binding. The effect of temperature on the enzyme substrate interaction or the effect of temperature on the Km value of the reaction gives you some insight into the thermodynamic state of the enzyme substrate binding, that what are the enthalpic and entropic parameters involved. One can analyze. Now, if you look at simply by your classical Van 't Hoff equation for a equilibrium process, I am talking of the equilibrium process is here E plus S going to ES, and with a dissociation constant of Km. Now, the free energy change will be equal to RT ln Km. Note, I have purposely removed the minus term from here, as Km essentially is not the equilibrium constant, is the dissociation constant, almost like inverse of that, and which again also can be equated to delta H minus T delta S. Of course, I think all of you may be familiar, conventionally in biochemical systems we are used to use this term with prime, and I am sure that you must have understood what is the meaning of prime for the thermodynamic parameters in biochemical systems. Well, anyway, that is only for the sake of convention, but essentially the laws of thermodynamics do apply, and the Van 't Hoff equation is also applicable, the second law of thermodynamics is also applicable, and therefore you can write ln Km as equal to delta H over RT minus delta S over R. So, a very simple situation arises that if you determine the Km values for an enzyme catalyzed reaction experimentally at a number of temperatures, and then make a plot between ln Km and 1 upon T, you will end up in a straight line with a slope of delta H over RT. Now, essentially the magnitude, so one can easily get delta H over R, not T. So, one can easily get the magnitude of delta H, the enthalpy change during the enzyme substrate binding. Also, the sign of the slope, I have shown only a positive slope, it could end up in a negative slope also. The sign also will indicate whether the enzyme substrate binding is exothermic or endothermic. You can get a thermodynamic picture of the enzyme substrate binding, and also if you notice that the interaction on the x axis will give you delta S over delta H. If you put this equal to 0, you get delta H over delta S as the interaction point, and thereby you can calculate both delta H and delta S for a given enzyme catalyzed reaction by studying the effect of temperature on the Km value of a reaction. The second effect of temperature in the case of enzyme catalyzed reaction is the dependence of K2, that is the turnover number or this the rate constant of the irreversible step of the conversion of ES into P, and that classically follows a Arrhenius kind of a pattern, that is your K2 is equal to A e to the power minus E star upon RT, where E is, we have seen this analysis even while we discussed in our initial lectures on catalysis. That means the catalytic pattern of the enzymes as a catalyst, and one can again analyze in the same manner. Ln K2 will be equal to ln A minus E star over RT, very familiar profile, 1 upon T versus ln K2, you get a negative profile, and this the slope of E naught E star upon R you get, and that gives you a magnitude of energy of activation for the given enzyme catalyzed reaction. So, the effect on K2, that is the turnover number, is primarily almost like any other chemical reaction. The third effect on the enzyme catalyzed reactions of temperature is the deactivation of enzymes. Just like in the case of pH effects on the deactivation of enzyme, temperatures are also mediated by a inactivation process. That means, as long as you are keeping the temperature regime in the environment of the reaction within a closed range, the enzyme may be functional. But if the temperature ranges are shifted drastically to the higher side, the deactivation occurs, and the deactivation essentially a very broad picture of deactivation, as we discussed earlier, is represented by a first order deactivation kinetics, where you can define as E is equal to E naught e to the power minus k d into T. That is your and you here also you can make a first order kinetics analysis, that is your ln E over E naught versus time will give you a profile with a negative slope, and slope will be equal to minus k d, and you can get the deactivation constant for the enzyme at now this essentially will be a function of temperature. Now, the magnitude of the deactivation constant will be a function of temperature. Now, one must look at the effect of the deactivation effect, the third effect, and the second effect, that is on the K2, as opposite forces. While with the increasing temperature, on the one side the reaction rate increases, while with also with the increase in temperature, the deactivation constant also increases, and there is more chance, and therefore the practical operational temperatures of enzyme reactions therefore are chosen on the basis of not just the optimum temperature, that means the optimum temperature for the enzyme reaction rate, but also in taking into account the deactivation kinetics, because the interest will be essentially on the total productivity of the enzyme per unit enzyme rather than only initial reaction rates. All these studies are based on initial reaction rates, but if you look at the practical application, that will be based on total productivity, and both the effects, the deactivation as well as increase of reaction rate with temperature, should be considered. So, to summarize, we have seen the effect of two major environmental parameters which are experienced by the enzyme catalyzed reaction. The first is pH. The effect is quite complex, and the effect can be considered on the basis of the shift in the equilibrium, or on the ionization of either substrate molecule or on the enzyme active sites. The other parameter, which is temperature, also influences in a multiple fashion. It influences to increase the reaction rate in a pattern of the Arrhenius plot. It also influences the deactivation of the enzyme molecule, and finally a very important tool which it provides us is to understand the thermodynamic status of the enzyme substrate interaction, and one can, by computing the value of Km at different temperatures, one can get a picture of the delta H and delta S for the given enzyme catalyzed reaction.